To talk about another interesting person.
Yes, there you go.
And that is our good friend and member Christian Schwarz, who I think is probably one of the 2 or 3 most knowledgeable people about what's coming up in this area, because he has spent a lot of time doing it. He is doing a number of other things. He has a book in progress that will be coming out. I had hoped any day now, but he tells me it's going to be any month, sometime after the first of the year. But it is going to be a remarkable collection of, it's going to be a definitive book on the mushrooms in California and the Redwood Coast. 750 species, he tells me, and that all involves pictures and then detailed both scientific descriptions and descriptions that are comprehensible to those of us who are philosophy majors. So we can look forward to that. And tonight, Christian and Adam are going to be talking about how it is that we all have the opportunity to contribute to the body of knowledge about the fungi that are coming up here. And I think the model is probably applicable much more broadly than that. So, Christian, welcome. Thank
you. First of all, I want to say thank you for having me again. It's really nice to hear Richard refer to me as a friend of the club, because I would much rather be that than just your speaker. We made it through another summer. The rains are just around the corner. It's not too long. Hold tight, guys. One thing that Richard said that I want to reiterate is that this is mostly not a talk about other things that I want you to learn, but about things that I hope that you will help me do and help each other do and help people who are like Tom Bruns at UC Berkeley do. So this is intended to get you thinking about what your larger role is in learning about mushrooms and advancing our knowledge in general about mushrooms. So for some of you who feel sidelined by all the recent changes that have gone on in taxonomy, it doesn't have to be that way. And I would like to show you some of the ways how. So I originally titled this talk Kicking and Screaming, Dragging Mycology into the 21st Century. But I realized last year didn't get very good turnout, and maybe it's because it was kind of a downer name. And part of my message later in this talk, I realized, is about us not, condemning ourselves. This doesn't have to be painful. So I decided to change the title for today. It's Skipping and Whistling, Waltzing Mycology into the 21st Century. This is going to be a walk in the park, guys. It's not going to be a big deal. Okay. Oh, I think David Rust might have sent it to me. Maybe that wasn't his name. Okay, so poll. Every year at the Fungus Fair, what percentage of mushrooms that are out on the tables are correctly identified? And let's use the strictest sense of correct here.
0.
It's not 0, thankfully. 50% is not a bad guess. 40% is a pretty good guess. Now, that's not as bad as it sounds, because many of the mushrooms are correctly identified to the level to which they can be correctly identified. Maybe the big elephant in the room that a lot of you don't know is that names we use are incorrect. And I'll talk about why those names are incorrect, or what it even means for a name to be incorrect, because they are fundamentally arbitrary when you get down to it. But a lot of the names that we use and take and share for, take for granted and share as a mutually understood name for a mushroom, which is what they're supposed to do. So they're functional and correct in that sense, but they're incorrect in a more scientific sense. And I'll talk about why that is. But it's a pretty dismal scenario if you look at it from a very strict perspective. Okay, so how many of those mushrooms do we have sort of a basic sense of their ecology and life history? Ecology we're pretty good at. We can tell this is mycorrhizal, this is a saprobe, that's a brown rot, this is a white rot. But life history gets a little more iffy. Is this a mushroom that makes a new mycelium every year and then dies? Or does the mycelium stay in the ground? Is it perennial or annual in the way we think of wildflowers as being perennial or annual? 20%. I don't know. I don't even know the answer to this question. But the fact is that it's a low percentage. We don't know very much about what goes on underground. And part of the reason is because it goes on underground. It's really hard to understand the life history of an organism that we can't see in our everyday lives. It's not like a bird that spends all of its time visible, in front of us. If we can find it, we can understand what it's doing. But with mushrooms, that's not the case. And then a last poll. What percentage of those mushrooms do we have documented geographic distribution maps for? Even the optimist gives you pretty bad numbers on this. So this is a very, very low percentage. We don't have good distribution maps for almost any mushroom, even the common ones, even the distinctive ones. These are the ones that are easy to identify and abundant every year.
That's because nobody here is willing to share where they find it.
Yeah, it's all you greedy porcini clutchers.
That is a crusade of mine.
Or it's because we're dragging spores all over the planet.
It's not just the edibles I'm talking about. I'll give the example of Amanita muscaria and how poorly we understand that mushroom a little bit later in the talk, but it's a low percentage. So these are all subpar scenarios. I would like for us to be able to identify mushrooms better. I would like for us to understand their life history better. And I would really like for us to have better distribution maps. But for that to happen, we got to address first things first. We have to be able to both recognize and identify mushrooms. And you'll notice that I say those things separately. Recognition is not the same as identification. Does that sort of make sense? Yeah. You all maybe recognize that there's this big orange sort of globular looking thing fruiting out of your eucalyptus every year before you were a mushroom person, and now you know how to identify it. But you could recognize that thing year to year. And these things are very closely tied together, but they're a little bit different. And I want to talk about ways that we can get better at both. So, how do you get better at recognizing mushrooms? Any ideas?
Practice.
Yeah. It's just practice. But there's many different phases of practice. So with mushrooms, unlike asbestos, long-term exposure to all aspects of mushrooms causes what? Yes, competency. So the idea is, no matter what you're doing, as long as you're in the presence of, talking to people about, writing about, thinking about mushrooms, you're getting better, even if you don't know that you're getting better. The more you're around the culture, the more that you're around the mushrooms, the more familiar you get with them, the better you get at every aspect of recognizing and identifying them. So familiarity is key, and that's, nowadays, that means lots and lots of time spent in the woods, at the bench, so back in the lab, writing descriptions of your mushrooms, looking at books, trying to identify mushrooms based on resource bases that we already have, the knowledge that's been published and written down. On the scope, if that floats your boat, so looking through the microscope, sort of getting to know the innards of your mushroom, so to speak, and online, doing whatever it is that happens online. How many of you know what happens online in a significant way for mushrooms? What are some of the most important things happening online? Okay, Mushroom Observer, someone said. We just got a new website for the club, and that's going to be a place that you can go to read Mushroom of the Month articles. And some of you might not immediately think of that as something that you do to get better at recognizing or identifying mushrooms, but I guarantee you, even doing little things like that makes you a better mushroom identifier, because you're thinking about them and you're learning what other people have read and thought about them. So a couple things that happen online I will get to a little bit later, but this is all fine. You can all do this all on your own, more or less, except for the online part. That's usually a communal sort of thing. But another thing that's really important is not just doing this on your own, but looking for mentors or being a mentor if you are someone who's experienced. And a lot of the people in this room are people who can be mentors to young people who are learning for the first time. So what you can provide as a mentor is validation. When someone gets a mushroom identification correctly, you can be there to say, yeah, you got it right, good job. Way to go, do it again next time. Correction, if they've misidentified a mushroom, especially a toxic mushroom, someone's got to be there to correct them. You can't just leave them to their own devices. And then feedback. Maybe they almost got the right identification or got the right identification for the wrong reasons. All of those things are important, and only really a mentor can provide that kind of service to someone who's just learning mushrooms. So online, Yahoo and Google groups, stuff happens. I really don't keep up on them as much as I used to for a number of reasons. But there is really good groups. Dimitar's, how many of you met Dimitar? Many of you have met Dimitar. He runs one called Mushroom Talk. It's a pretty popular Yahoo group. I don't really read it anymore, but a lot of good stuff goes on there. It has its phases. BAMS has one. This is, I believe, the North American Mycoflora Project's Yahoo group. Not much goes on there yet because it's pretty new. But a really important place for you all to know about online is mushroomobserver.org. How many of you have been to this website? Was there anyone who's not been to this website? If you are interested in mushrooms, if you're interested in learning to identify mushrooms, if you're interested in helping other people correctly identify their mushrooms, this is probably the best place on the internet right now to do all of that. It's a little bit chaotic. It's not a very clean website compared to some places that get better funding, but it's really the best we've got, and it's really actually quite good at what it does. So the idea here is that you make a profile and you upload photos of what you found, and you propose names for them, and then people will say, oh, I definitely think it's not that, or I definitely agree with you. And after the community votes, it's quite often the correct identification. Sometimes it's a little bit tilted, but it's getting better. Unfortunately, it's not really sorted through for quality control yet. There's no sort of verification, but we're working on getting that sort of making that be part of the base functionality of the site. But so far it's not that way. And it's global. So you'll see mushrooms from all over the world. If you take a vacation in Hawaii, you can upload the mushrooms from there. If you go to South Africa, you can upload mushrooms from there. If you go to Antarctica, you could upload the first record of a mushroom from Antarctica. Because so far they've got 0. Oh no, that's not true. My friend Kira was working down there as a carpenter, and Noah, who some of you have met or seen speak here, asked her to put a piece of bread under her bed while she was there and photograph it when she left, and she uploaded that to Mushroom Observer. So we do have fungi from Antarctica now. There's people on there who have ringworm infections. You know, there's a little photo of their side. But Mushroom Observer, resistance is futile. You should join. Okay. Yeah, we're just trying to make the quality of life better for all species. So, learning to identify a mushroom. This is matching a face that you've become familiar with to a name. So you might be able to recognize the face, but until you learn its name, you can't really identify it. Identifying it gives you a tool to communicate to other people with. It's an agreed upon handle for that thing that we all recognize as this. So, a really good place, I think, to start for mushrooms is genus level. Different areas of the tree of life have different good entry points. So with plants, if you took systematic botany in college, you probably studied families. If you took ornithology and you studied birds, you probably went straight to species because they're pretty well characterized and readily identifiable. I think with mushrooms, and many of you know this, genus is a really good place to start. It breaks them down into sort of compact groups that you can learn to recognize not only in California but across the nation. It gives you a really good sort of library to start with. And then you can learn species later because there's many, many more species of mushrooms than there are, say, birds. Okay, so species comes next, but that's a little bit more advanced usually. You'll learn this in stages, but genus is a generally good way to start. So with birds, like I said, you start with species, plants, families, and then mushrooms. I think there should be an emphasis on genera. So keep that in mind if you are helping someone learn for the first time, say. So I would like to emphasize this, but there are some stumbling blocks to learning to identify genera. With the recent advent of DNA technology and our understanding of phylogenies, which is how organisms are related to each other, we have decided that if two organisms are close enough to one another genetically, no matter how different they look, we have to call them the same genus, say. So this little knobbly truffle thing grows underground, no gills. It looks like chewing gum, old chewing gum. And this beautiful Russula that grows in our Douglas-fir forests would probably have to be in the same genus, Russula, from a phylogenetic standpoint. So This makes it difficult to teach someone what a russula looks like, doesn't it? It's just, a beautiful normal mushroom with gills and a stem and a cap, unless it's a gnarled marble. That's not exactly a cohesive message to give someone. So there are a few stumbling blocks, and they're still being worked out both in the academic community as well as on the teaching side. But I think we can find our way around them. It'll just be a slightly more complicated picture. Yeah?
Out of curiosity, will those not be the pink crucibles every year, or will they be the non-meat mushrooms every year? Or do they switch back and forth between?
It would be fascinating if we found that out, but I don't think there's any evidence suggesting that they spend some years as a Secotioid fruit body and some years as a gilled fruit body. I think that's just their growth form. These grow in the Sierra, these grow on the coast, and there's really different environmental pressures there. So in the Sierra, they have these big swings in temperature and ambient humidity, so it makes sense for this mushroom to grow underground. Yeah, it keeps its spore-producing surface away from the dry weather in a more stable temperature and humidity. So it gives the spores a better chance of surviving. Okay, so if we move on from genus and go to species, we face another set of problems. How many of you have felt this? The names are constantly in flux. The names are always changing. Okay, I have two messages about this. One, it is not your fault. Two, it's not going away. You're going to have to learn to deal with this. But it can be made easier for you, and we can make it easier on ourselves. Part of the problem is that not only are they in constant flux, there's the lack of a referee. There's no one saying, okay, now this new name is here, let's all start using it. There's all these varying interpretations where some authorities accept other authorities' names and some hold bitter personal clashes and refuse to accept each other's names, which is ridiculous. It's not that way in other areas of the tree of life. For example, in birds, there's a committee where they say, all right, even if we don't like it, this is our consensus right now, and we're going to use this name, ego aside, mostly. So, yeah. Another problem is that we've done a lot of borrowing. We have taken European names for mushrooms and applied them to mushrooms that look similar and may even be closely related, or they might not be at all, to mushrooms that grow in the US. And in California especially, that's a bad idea. So, lack of a referee results in situations like this. Inocephalus, Puzarella, Alboleptonia, Inocephalus, Paraexilia, what is this? Exilia, Clytopolis, Trichopolis, another Inocephalus. Or they're all Entoloma. There's literally two different camps on that. You could call them all Entoloma, or you could memorize all of those. But no one has decided which we're going to go with for the time being. It's just people working independently, which, as you can imagine, makes for a very jumbled and difficult to interpret literature. You'll read in one book Entolomas and have one idea in your head, and read in another book all these different names, and you're not really sure if they're talking about the same organisms when in fact they are. So it'd be great if we could figure out some sort of refereeing system for this. Okay, we also have to stop borrowing. This is Lactarius deliciosus. It's a European mushroom. It's been introduced to other places around the world, but not here. This is what we call Lactarius deliciosus, and they are not the same. They're not even the same, they're not delicious by most people's count. Some people like them, but these are supposed to be really good. These are supposed to be a very sought after mushroom. But you'll notice Lactarius deliciosus in the true sense doesn't turn green. It doesn't. It's not part of the original description of that mushroom. And ours consistently turns green. So there's these even clear visible differences that we've decided to ignore for whatever reason and borrow the European name wholesale. Now, why is this such a particular problem for California? Well, look at the climate zones. We basically have our own climate zone. This is the California climate zone, more or less, which means that a lot of the organisms that live here are not like the organisms that have made their living throughout the rest of the country, much less Europe. So California is a land of endemics. How many of you know the word endemic? So endemic means restricted to one place and only that place. What's a good example of this? Redwoods are endemic from Monterey County to 6 miles over the border into Oregon, and that's the only place they live in the world. So they're endemic to that part of the coast. Say that again? Someone says it? Okay. Okay, so another thing that we need to do to stop the constant flow of names is actually just make one big push to change a bunch of names at once. So this has the very biblical sounding name of Nomenclature Resurrection, but I promise it's not that scary. There's no bloodshed involved. Well, mostly. Yeah. But what this involves is comes from a couple basic principles of how we name organisms. Every organism is supposed to be named based on a type collection. So what that means is you find a mushroom that's new to science, no one's ever named it before, and you collect those mushrooms and dry those mushrooms, and those fruit bodies are the ones that that entire concept of that species is based on, which is kind of a, it's a stressful situation for that mushroom. It's got to represent its entire species. Fun fact, there's no type specimen for humans, but Phil might be a good one. So a type carries a lot of weight. If a mushroom is not genetically identical, if it's not microscopically identical, if it doesn't match the physical description of the type, it doesn't get to be called the same name as the type. That's how this is supposed to work. Priority. If two mushrooms were called two different names, whichever has the older type has naming priority, even if it's a stupid name, unfortunately. So priority rules come into play here. If there's an earlier name for the same mushroom, that's the name we're supposed to use. That's the rule, no matter how much we would like to keep the new one. Synonymy. If two mushrooms are found to be the same mushroom, even though they have different names, you have to collapse them into the older name. So that's called desynonymizing. Okay, so what we need to do is, does anyone see how this relates to our situation between Europe and the US, Europe and California? Remember that there was no internet for a long time, and mail took forever. And that there were people working both in Europe and in California at the same time. But what did that allow them to do? Or what did that result in?
All of our mushrooms became European names.
Well, we may have adopted European names, but the cool thing is, before there was free communication, a lot of them got named twice. A lot of them got California names and European names. And what we have to do is go back through and figure out which ones actually do have California names and resurrect those. That's what the nomenclature resurrection is about. Bring those back into common usage. Then no one has to do any work. You don't have to re-describe the mushroom. It's already been done. You just have to start using the appropriate name. And many of these names were forgotten in favor of the European name because the European name was more well characterized and more well known in the academic community. So how many of you know the cedar waxcap? What do you call it? Camerophilus russo-coriaceus. Means Russian leather. Smells like Russian leather apparently. I don't know. There's actually an existing name for that mushroom, which is going to escape me for the moment. But it was described from Oregon, I believe. I think it doesn't matter if it was earlier because we know now that they're different mushrooms that are similar. So you don't have to get rid of that old name. You just can only use it when you're in Europe. But we have a name for this mushroom, and we can resurrect it out of the books, dust it off, and use that name because it's a good name for the West Coast. It describes our mushroom, not the European one. So, to do. Make a list of those species that have types from the West Coast and compare those mushrooms to the European counterpart. Establish actually that they are different, and then start using that name again. That's all it takes for many of these mushrooms. They've already been described. We don't have to do the work. Okay, so learning to identify species, we talked about constant flux. Another reason for that has been DNA sequencing. We've learned that mushrooms that look really similar are not the same. Yeah?
When you look at a mushroom like, say, Edulus in
Yeah.
Europe, and you look at an Edulus here, how much different does the DNA have to be before you can say, okay, this is a unique species, and that's different from that species, therefore we can use that California name just describe that thing and say yes, okay.
Yeah.
It's sort of like the chanterelle, for example, on the East Coast is tiny,
Yeah.
and on the West Coast is big, and that sort of thing. But I mean, boletes and that sort of stuff,
Yeah.
we just say, well, Is it 90%? Is it 75%?
Yeah.
You know, how close to that?
There are some rules of thumb, but there are no hard rules because it's literally arbitrary. These are not natural standards. Mushrooms don't meet and compare their DNA and decide whether or not they're going to mate. So we are forcing this system on them, but it's up to us to interpret the results of our investigations gracefully. Yeah.
As you're doing the Nomenclature Resurrection, how are you communicating this to the rest of the community?
I will talk about that in a second. That's a great point.
In scholarly journals, essentially, and putting that out there. That's
Which-
your reviewed journal.
Yes, but it doesn't have to be that way. And I don't think it should be that way. But I'm glad you brought that up because I will definitely be talking about that. So to answer your question, Richard, you have to just set up a bunch of criteria, present your argument, and hope that the people who are in charge of determining consensus will side with you. And some people are really good at doing that, and some people are not good at doing that at all. Some people will take a very weak, a very tenuous difference in DNA sequence and decide that they've got enough ammunition to name a new mushroom, and frequently do, and don't get a lot of pushback. No one challenges them on it because it's honestly a pretty small academic community. Maybe you didn't know that. Mushrooms aren't the forefront of science every year. So in any case, DNA sequencing hasn't just been more information, it's a new kind of information. It's totally divorced from what we can see with our senses. It's not something that has any sort of bearing on the mushroom's daily life biologically. It turns out that the area of DNA that we sequence, for the most part, is not functional DNA. It doesn't do anything. It's sort of filler. It's spacer DNA. So we really have to learn how to interpret the results of our investigations carefully, better. As we'll see in a second, it's not always been done. There has been very ungraceful interpretations of genetic data. Lindner et al. Studied Laetiparus. You all know the sulfur shelf mushroom? They took ITS sequences, DNA sequences from those mushrooms across the US, and then they did mating studies. And mating studies actually do matter, because what does that mean? Mushrooms can produce fertile offspring, which is at least one person's definition of a species. Yeah? The biological species concept states that if two organisms are interfertile, they belong to the same species. And that actually has some biological relevance because they can mix DNA and be subjected to the same evolutionary outcomes, whereas their spacer DNA has nothing to do with that. So they compared the results of their mating experiments and their ITS sequences, and they found that the ITS sequences suggested that there was maybe like 15 different species, where their mating studies suggested that there was only 6. There were only 6 interfertile pairs. So what they found is that the ITS overestimated the diversity significantly. How widespread this is within fungi, we don't know yet. They haven't done similar studies, and in some cases, studies are impossible. It's really hard to do mating studies on ectomycorrhizal fungi, like the boletes and the chanterelles, because you have to grow a forest to culture them. Laetiparus you can do in the lab because they're saprobic. You can just feed them wood chunks. So yeah, we have some refining to do. So, what was your name?
Jason.
Jason brought up the publicity of these names. So when a name gets changed, what happens nowadays? It gets published in an academic journal. How many of you have access to academic journals? Those of you who work at a university? I used to. That's great. But I can't even get a copy of my articles if I go to Mycology. I have to buy them from them. Thankfully, I have them on my computer because I wrote them. But it's not easy for the average person to get access to this. Yeah?
Cal Berkeley Science Library is open to the public.
You can walk into a physical library. Then you have to hope that they have a subscription to the journal. UCSC just got rid of their Mycology subscription. I couldn't believe it. It's the, what's that?
They cut way back on all of it.
Yeah, it's horrifying. I'm like, there are people here who study fungi and they now do not have the leading journal on fungi.
Did they cut the electronic connection to it, or did they just not?
Do you know, Wendy?
Physical?
The library, I don't know specifically the library is.
I would have a hard time believing that they cut the electronic one.
That would be.
Yeah. But what it means is you can't go in and see a physical copy. So you have to go to Berkeley. which is, I'm not a fan of this system. Your tax money goes to the NSF, which funds some of the studies that produce the articles that get published in these. It seems like you should have some sort of access to them, especially when they're not commercially important. This is about diversity, not about, I don't know, new petroleum derivatives. So there are alternative options. Many authors can now request that their article be freely available if they pay a little bit of a premium, which I know Dimitar does on all of his, which is amazing. He pays extra so that we can get it free. I wasn't able to do that because it was pretty expensive, but I've been posting it places for free. You know, not sure if I'm supposed to do that, but oh well. But there is a number of journals now that are only online and are accessible freely. How many of you heard of North American Fungi? It's done out of the Pacific Northwest. It's a great, great publication. It's online only, but anyone can access it and anyone can submit to it. You still have to clear peer review, you still have to get it reviewed, but you can submit an article there. And I'll be talking about this later, but I encourage you to consider that if you've got something you would like to write about. Even if it's a basic study, you can do it. Okay, so yes, we have this lovely thing called the internet. It's been around for a while now. And yet we still have things like the Morel taxonomy fiasco of 2012. How many of you heard about this or felt its impacts?
Still not over it.
Yeah, no. I still stay up nights. A bunch of new Morels were described after years of study, years of soliciting collections from people all over the United States. Michael Kuo, some of you have seen him speak over in Illinois. He was doing this, painstakingly gathering information, published his paper, and found out that 2 days prior, someone named Cloez had published in his Mycological Society's journal in France, print-only French journal, some of the same mushrooms that he was working on. And they had failed to communicate completely. But because Michael Kuo's preview came out 2 days earlier than Cloez's, it's uncertain who has priority. Now, there's a couple of problems with this. One is that there was just no communication going on, and they could have avoided stepping on each other's toes and making a mess. And two, Michael Kuo was describing mushrooms he'd never ever seen in person. And Cloez was describing mushrooms that he had definitely never ever seen in person. He was getting morels out of people's freezers and describing them from across an ocean. So he gave them silly names like the swarm of bees morel, because they thought it looked like a beehive, which, they sort of do. But it was based on one frozen specimen, and that was as much familiarity as he had with the mushroom. Whereas Michael Kuo had a really robust species concept and a good name for it. So these sort of things don't have to happen because of the internet. It's a really basic principle here. So I think we could set up sort of repositories for people to talk about what they're working on, collaborate rather than just sort of get in each other's way. Because this is a headache for both of them now. It's not like one of them is the clear winner here. So yeah, we can avoid this. Cameras that produce digital files, in color even. These are common. Everyone's probably got one in their pocket. Many of you do anyways. I don't actually have my phone on me, so I can't say that. But there's There's 5 cameras pointed at me right now. There's one on the computer, there's one on the phone. We can all take colored pictures of mushrooms and send them to each other so that we know what we're talking about, more or less. There are still publications coming out in academic journals describing new fungi without colored pictures attached to them, which is horrifying. It's just backwards. It's ancient. We can do this. It's not that hard. And then, yes, put them on the internet so we can all see them. I was told that smell-o-vision is next. Apple had that tech conference recently, the iPhone 5. This is their newest model. Oh, no. It's back.
Not that one.
This is called the eye stink. This one will really help you identify it. Yeah. So, learning to identify species, group identifications. Identifications done sort of by consensus, by a number of people working together and deciding, oh, I really don't think this fits, or yeah, that's, dead ringer. That's definitely what it is. I think this should be more commonplace. A great way to get this done is through table talks. When you go to a fair, attend the table talks. Go see what people have to say about the mushrooms. This is like when Phil or Henry walks around a table and picks up mushrooms and talks about why this one is identified as this or why this one is definitely not a chanterelle. Attend those, listen to them, and if you think that maybe one of the mushrooms on the table is misidentified or you have a question about why it's not what you think it is, ask. I think that collaborative identification should happen a lot more often. Unfortunately, that means you have to, well, not unfortunately, but that means you have to bring people up to a level where they can communicate sort of on the same plane of knowledge, which means we're going to have to do a lot of pickup work with a lot of people who come to the fair because they don't know anything. But this is not the case in, say, Europe. A lot of people there are on the same level of being able to identify mushrooms, and they can disagree with each other in productive and intelligent ways that lead eventually to realizing that, oh, we've been calling two species one name for years. But I noticed that this one doesn't stain blue at all anytime I've ever collected it, and it's associated with pines. And the ones that stain blue are always associated with oaks. There's two different boletes here. So it's an important thing to share your thoughts about why an identification might be correct or incorrect, and I urge you to do it. And I bring you good tidings, sort of. Everyone is wrong about some things. This is true. Everyone is wrong and ignorant about other things, which means you don't even know you're wrong. And this is probably the more common problem in fungi. In mushrooms, this includes the experts. People who make their living as professors teaching mushroom taxonomy are frequently wrong and frequently ignorant that they're wrong. And there's reasons for this. It's not really their fault, because being a professor is not a good way to structure your life if you want to get good at identifying a bunch of esoteric organisms. Because you have things to do like mentor students and teach and do paperwork and be a professor at a university. Natural history sort of takes a bunch of time, and it can be difficult to balance those two. So frequently they know that they're wrong about most mushrooms, and they will defer to an amateur who spends a lot more time in the field to identify their mushrooms. And you should know that. You should not feel like when, say, Gary Linkoff is in the room, although he's actually he knows a lot of what he's talking about. Tom Bruns can identify a fair amount of mushrooms, and he knows it. And that's why he comes and asks people in the Fungus Federation, hey, what is this? Now, if you want to talk to him about crust fungi, he knows more than all of us in this room will ever know. But frequently experts are experts in a very small group of fungi. And so you shouldn't feel intimidated, and you actually should feel like you have some service to provide them, because you have a broader knowledge of mushrooms. Okay, so as long as this doesn't involve edible and poisonous species, everything is fine, and we don't need to feel like this is an ego issue. Now, if it does involve edible and poisonous species being confused, then we need to get a little testy with each other and make sure that no one gets hurt. Okay, so sometimes it's up to amateurs. The experts can be especially wrong because they don't spend time in the field. The United States is a huge area to cover. If we're going to do a North American Mycoflora Project, it is not going to be primarily done by academics. It's just not. It's going to be done by people like us. People who are out in the field maybe every week, maybe 2 or 3 times a week during the season. Maybe we go somewhere during the summer. Maybe we take a vacation and do mushroom observer observations while we're, say, in China or in Mexico, or even on the East Coast, or maybe just down in San Diego. All of that is important. All of that contributes to what a mycoflora is. And I'll talk more about what exactly that means in a second. But we have a huge area to cover. And we have many, many, many species. This is not like birds, where 700 species or so have been recorded in the United States. A little over that now. There's many more. There's 700 going to be, I think I have like 1,300 species from Santa Cruz County that I've seen. What does that mean for California? No one knows. No one knows what our upper limit is here. We don't know how many species we have in California. And frequently, increasingly, taxonomy is de-emphasized in universities. So we can't rely on a new generation of academics to help us out here. Really, the way the tide is turning is for taxonomy to be done really, really well by amateurs and enthusiasts. And what that means for experts, academics, is that they dovetail their efforts with ecology and sequencing with our ability to identify mushrooms in the field and our knowledge of where they grow and how they behave. Okay, so let's assume that we all know how to identify mushrooms now, and we can identify every mushroom we see. This is a wonderful, wonderful world we now live in. We move on to ecology. Where do mushrooms grow? What do they do? Distribution. Where in space do they grow? Where don't they grow? That's just as important to know. And their status. Are they endangered? Are they frequent? Are they abundant? Or are they just barely hanging on by the skin of their teeth? Are they really sporadic? Are they endemic to a region? apparently included this twice. Oh, life histories. So we want to know more about how they partner with their tree and what they do underground. Distribution, we want to make maps. So what that turns out, what that will look like when we have assembled a good distribution for a mushroom is a map. You'll see a bunch of red dots saying this is where we found this mushroom, and we've sampled the area over and over, so we're pretty sure it does occur here every year, and we're pretty sure it never occurs here, no matter what year you go there. And then status. This is really the only way that we could get a mushroom, say, on an endangered species list, is if we have really good status data. How many of you know of an endangered mushroom in the West?
What?
Is there an endangered mushroom in the West? Yeah, there's a shell fungus in the Pacific Northwest that grows on grand fir, Brygioporus nobilissimus. It's an amazing mushroom. I've only seen one ever, but it's like 5 feet wide, and it's white, and it's got hairs on it that stand that far off the cap. So you can like run your fingers through it. It feels like I don't know, mohair almost. It's amazing. And it's really pretty rare. There's, I think, like 85 known fruit bodies, something like that. So it's quite rare. It's endangered.
What about Agaricus?
Agaricus is actually much more common than is thought. How many of you know what Agaricus is? Fomitopsis officinalis, very medicinal mushroom. We got our first records for Santa Cruz County this year. It grows here on big Douglas firs. But up in the Pacific Northwest, it grows on other conifers. So, all of those things that I just mentioned, identification, status, distribution, and ecology, those are the pieces of a flora, or a mycota, or a fauna. If you're doing an avifauna of an area, you want to know all of those things about the birds of an area. So we'll call ours a Microflora. So we need to catch up, because in birding, they have a public database called eBird that citizen scientists, you and I, can go out tomorrow, see a bunch of birds, write them down, submit them to this database, and they'll become integrated into this dynamic data set that professors are using to publish their own papers now. And they always give a shout out to all the people who helped them collect that data. Yeah.
Is there no screening? You can just dump. What if you're dumping?
There is lots of screening.
Okay.
Yes. They have a lot of money. So this is a massive ongoing citizen science initiative. I just entered an eBird checklist yesterday for things that I saw. This data is used in scientific publications. There's many, many publications now. The first one, everyone was like, oh, this is amazing. We've finally done something. And then within the next month, there was like 15 more. So as soon as people realized they could use this data, they started using it in a big way. And we can do that for fungi. Of course, there are some things going in their favor. Every North American species of bird has been discovered. They're all known. At this point, they're just splitting species. Subspecies can be identified in the field. That's not something we can do yet with fungi. And they're on to sorting out evolutionary relationships. So towards the end of a North American Mycoflora, Berkeley, Yale, I forget what FFSC stands for, Fungal Environmental Statistical Informational Network, I don't know, something ridiculous. They had this conference last summer. Yeah, last summer. There you go, Environmental Sampling and Informatics Network. You know I can't read, Bill.
I just figured that out.
They decided to have this conference to discuss the beginning of a North American Mycoflora project, because it turns out that no single entity, no state, no county, no region, and certainly not the United States as a whole, none of those entities have ever compiled a full Mycoflora. There's never been an exhaustive list compiled for any region in the United States, which is pretty pitiful. So they decided we should change that. So they want to make a list of all the, not plant, that's a flora, but so for plants, it's all the plant species known to occur in an area, the diversity, and it's often a flora is designed to provide some taxonomic clarity. So it's designed to say this does occur here and this doesn't, even though they're very similar. And so if you're in this area, you know that you're looking at this and not this. So it's sort of an identification handbook, but not really. And then the distribution and status of those species. Now, I think Santa Cruz is a very special place to be compiling one of these. Now, why is that? Santa Cruz has a huge species to area quotient. Now, we're a little spoiled in that regard. Does anyone know why? We're the second smallest county in California, so our divisor is very small. But that's not the only reason. We are special. we have endemic habitats, a lot of endemic habitats. Redwoods are endemic to the west, just the coastal west, and not even all of it. And we also have things like the Zayante sandhills. That habitat is entirely endemic to Santa Cruz County. So there's insects, plants. I believe there's a subspecies of ground squirrel or chipmunk or something that's endemic to the sandhills. There's lots of things that occur here and nowhere else. The same goes for fungi. There's lots that occurs in our general area, the Redwood Coast, and in Santa Cruz County that don't grow anywhere else. So this is a special place to be attempting to put together a flora, Microflora. Now we have plenty of floristic studies in California. The Jepson Manual, if you've ever used it, is an awesome reference. CalFlora is an all-in-line version of the Jepson Manual. You've got lots of places to go to if you want to study plants in California. But for mycota studies in California, you have no options. You're singing with the crickets. What we do have is an outstanding resource. But this is not, believe it or not, a Microflora. Why not? Have you looked at the Leptonia section? He explicitly says, I am ignoring most Leptonia. That's not the idea of a flora. A flora says, I know this group is tough, but I'm going to tackle it anyways, because a flora is intended to be totally exhaustive and comprehensive. So we have some challenges. The mycelium is not the fruit body. What we can see above ground is not what occurs most of the year. So we could go to a place where a mushroom grows and not see it because we're there at the wrong time of the year. That's a problem with some of the annual wildflowers, but it's not usually a problem for plants. Mushrooms go away really quick. They might fruit and they'll rot and we miss them. We have a lot of taxonomic ignorance. There's a lot we don't know about identifying yet. And we have been our own worst enemy. We fail to take notes a lot of the time. How many of you go on walks in the woods and identify a bunch of mushrooms and don't write them down and tell someone about it? Yeah, I know. We all do. We all do. But we don't have to be that way. People who study moths have it way worse. These things fly around at night. They fly away from you. There's thousands upon thousands more moths than there are fungi, and they have put together these awesome websites. So we can't be like, oh, woe is me, mushrooms are so hard. At least it's not moths.
Don't run.
How many of you can identify this mushroom? How many of you can find it every year? How many points are on the Mushroom Observer map? This is grim, people. This is awful. You can go out and double the number of points on the map in one day if you felt like driving that much in Santa Cruz County. So we need to be taking notes and uploading them. It's pretty easy, it's pretty simple, and it eventually becomes really rewarding. If you have someone willing to take all your data and host it for you and organize it for you and make it searchable, when someone asks you, where did you find that blue albatrellus all those years ago? You say, I don't remember, but I can go online and look up my own record and it'll tell me. And if you've added notes, you get to store all of that outside your head. So you can think about things like, children and going to school and feeding yourself. It takes up a lot of my mental space. So to that end, and Adam will be talking about this a lot later, we're trying to pair Mushroom Observer, which is a database, with something that's a little more of a resource that dovetails with the database. So the Mycota of Santa Cruz County is going to be, well, it's already live, but don't go there yet. We're not quite ready. But it's going to be the place for you to go to learn about the taxonomy of mushrooms in our area and to get an exhaustive list. Not just the easy ones to identify, but also the devilishly hard ones to identify. Things that we've probably misidentified will be on there. Hopefully not too many of those. Okay, so we want to make maps. I'm really interested in making maps of mushrooms. I actually do spend time awake at night thinking about this. But you can help by making your own maps. No scale is too small. Make a map of your yard. Make a map of a state park you live near. It's probably better to start at a very small scale because you'll be able to do it better. It's really hard and time-consuming and expensive to drive around California making maps of mushrooms, which is why we leave it to Noah Siegel. Organize regional initiatives. How many of you went to the Myco Blitzes that Berkeley and MSSF did out on Point Reyes? They did these awesome things where they got a bunch of people like you and I out into the woods, collecting things, putting slips on them, bringing them back, and then they had academics descend on the pile of mushrooms and identify as many as they could, and they got really, really awesome data out of that. And they have a really good list for the park. It's almost a flora. They almost have an exhaustive list there. Yeah?
Have you ever, do you know about iNaturalist?
Yes.
I'm really intrigued with it and want to start using it, but I don't have a cell phone. But anyway, I didn't know if they didn't have much input on mushrooms.
They have people contributing mushrooms. They don't have many people identifying mushrooms on iNaturalist yet. Hopefully, so my problem with iNaturalist is that it's basically just Mushroom Observer for everything. It's Plant Observer, Insect Observer, which is awesome. I'm actually glad that there's a clearinghouse. But what I would love for them to do is to use each other's data so that we don't have to do things twice. Have all of the Mushroom Observer observations be naturally sucked into the iNaturalist database and vice versa. So I think they need to learn to play together because I don't want to be entering data 2 times when I could be doing it once. But I think it's actually becoming very popular and really well attended. So I hope to see it take off more as long as they work together eventually. So the reason the MycoBlix worked is that they had tons and tons of volunteers. They clearly defined their area. They didn't just say, you know, all of the West Coast or California or even the Redwood Coast is a little huge and unwieldy. They said 1 state park, Point Reyes. And so if you were a step out and you found a new mushroom, it didn't count because that's not what a flora is about. A flora is valuable because it's arbitrary. You're saying only the ones that have ever been found inside these boundaries. So someone who's within those boundaries has a more useful piece of information than just sort of a general West Coast guide to mushrooms. And then they brought in expert taxonomists from all over to work on the vouchered, photographed, continually researched. The identification went on for months after the MycoBlitz was over. That kind of sustained effort is critical for making a mycoflora, but it's hard to do. So it takes some organizing, but it can be done. Okay, so the North American Mycoflora Project has this website. It's a little bit uninspiring, both visually and in terms of its motto. Without a sequenced specimen, it's a rumor. I don't believe that at all. I would encourage you to not believe that with me. I've sent them angry emails. Hopefully we'll change it. So why does that make me angry? Does anyone want to hazard a guess as to why that makes me angry? Without a sequenced specimen, it's a rumor. That's their motto. How many of you have ever sequenced a mushroom? How many of you know that Blvd sedulus grows in this county? They're disempowering basically everyone who doesn't have access to a sequencer, which is going to shoot them in the foot. You're not going to get a flora done if every single specimen you have involved in it has to be sequenced. And it doesn't have to be that way. Do you think every time that a new bird is found in Santa Cruz County, they go have someone shoot it?
Yeah!
It's actually not the case. It's just not the way that you have to do science. I think it's pretty ridiculous. So a way to get around that is have regional compilers, people who are familiar with what's local, what grows locally. So if someone says, how many of you know what Blvd frostii is? It's this beautiful eastern red bolete with a cherry red cap and stipe and stains blue and never, ever, ever would be found in California, probably. But a regional compiler's job would be to say, if someone said, oh, I found Blvd frostii here and added it to the database, your job would be to either nail down the identification, ask them to provide you a specimen, or chuck it out of the database. And this is how eBird does what it does. It has volunteer regional compilers go through the data, and they have an automatic flag. So if it's something that hasn't been recorded from the region before, it automatically brings it to their attention, and they either get rid of it or explore, find out if that's actually what it was, and either keep it in the database or take it out. So it's another way of preventing information loss. So if someone tells a regional compiler, oh, hey, I found this here this day, they may not enter it in the database, but the regional compiler does. So the regional compiler's job is to prevent information loss. It's also quality control, like I just talked about with Blvd frostii. It's making sure that incorrect identifications don't get incorporated into the database. One way this happens is through listservs. So if people type up a trip report, a compiler's job would be to add those mushrooms to the database, even if the person who wrote the trip report didn't. These are just some things to think about how a flora would be structured, or the compiling of a flora would be structured. I've mostly said all this. Oh, the database should be public. So all of the data that's generated can't be just squirreled away somewhere on a server. It's got to have some front-facing, public, yeah.
Kind of like Google Earth, when you take a photo
Yeah.
and upload it, it's location specific.
Yep.
So it's really big detail. I wonder if there'd be somebody at Google that'd be a micro geek that would want to.
I think we know someone there.
I mean, because if it's already a service that happens with the location services of photos, I mean, then I'm out there in the field taking photos and I don't need to take the notes on the maps.
Yeah.
Right there.
Yeah.
Focus on the description of it instead.
Yeah, there are applications for my smartphone where I can enter a list for birds and it will automatically find my location and reference it there in the database. So it's a little bit less work that I have to do. And all of that, is doable. We just have to make it happen. So something that a database also should do is to highlight the rarities, interesting abundances, and interesting absences. Last year was an awful year for chanterelles. A database should take all of the data relevant to that and sort of make a story out of it. Say, chanterelles had a terrible year, but some bizarre rare bolete had a great year. Or a new species was found in the county that had never been found before. I have 2 specimens of Agaricus americanus in a box up here brought to me by Ron and Henry. That was the first record for our county this summer. It was fruiting this summer. So it never stops. New things are found frequently. I think Britain gets an average of 3 new fungi for the island every year now, on average, which is remarkable given how long they have spent studying the Microflora of the British Isles. So where does all of this get publicized? Well, we can have an online database. We can also have amateur publications that are in print. Many European clubs have print publications. I'm going to pass them around. They are Czech, but they translate them into English for no good reason. So if you look through, there's both Czech and English articles in here. One of them is from the British Mycological Society, Field Mycology. All sorts of neat stuff in here. Some of them are single mushrooms, a single mushroom that's new for, say, a state park. They'll publish an article on. Very simple article. Doesn't even need to be peer reviewed because it's not particularly important. I mean, the editors will have a look, and if it's clearly bogus, they're not going to publish it. But they're really nice. They're simple. They're small. They're cheap to print, and they get actually pretty big subscribership to them. And it's not all technical. Some of it is just nice photography. So I'm passing a bunch of those around, but I'm also passing around some that local bird clubs do. These are also really cheap. They're not fancy publications. And in these, a really interesting thing that happens is that people will do a study. They'll just count the number of turkey vultures roosting in a particular tree from year to year and do a 10-year summary. And to say, we had an unusual low number of turkey vultures in 2011. We think it may be tied to the destruction of some of their foraging habitat over here. But I'll pass some of those around. These are all amateur publications that I'm passing around. None of these have any tie to any academic body, which I think is really cool. And the mushroom ones in particular are lovely. They're really nicely done. Okay, so the CVBS Bird Club, that's going around. The European clubs are going around. They publish research. They publish new records, including photos. So if a new mushroom is found for a particular area, they'll just publicize it in not only their online publications, but in these print publications. And the reason I think this is important is sort of came from what I learned when I was in the education program at UCSC. If you create narratives, if you tell stories about these things that are otherwise pretty esoteric, it helps with scaffolding in the human brain. We're good at telling stories, and we understand things through stories and the narratives we build around them. So if we can make the idiosyncrasies of a season, what had a really good year, what had a really bad year, if we can make that into a story, if the identification, the discovery of a new species can be made into a compelling story, it'll help people be interested in it and understand how it works and maybe do it themselves. Same for identification challenges. If it's not a new species, but you tell the story of how you went about identifying a particularly hard mushroom, that can be an interesting story that compels people to learn how to do it on their own. Yeah, so what this is called, another thing I want to talk about with narratives is self-fulfilling prophecies, especially negative ones. If we tell ourselves that we can't do something, we frequently get in our own way in subconscious ways towards that goal. So particularly towards Romeria, Cortinarius, Mycena, and Russula. A lot of people say, I just can't identify those. And if you say that, you're probably never going to be able to do it. Yes, these are difficult groups, but I think we should stop saying that they're impossible groups. We act like that a lot. I mean, think about it. When you're at the fungus fair, I mean, we'll still act like this. It's going to take a while. But when you're at the fungus fair and someone brings in a box of Cortinarius and Russula, you say, we'll get to that later.
Yeah, we do somebody to identify.
But in general, I think we should quit being so defeatist about it. Most Cortinarius and Inocebi in Europe are identifiable. A little elbow grease may be necessary, but they're worked out more or less. New Zealand has almost all of their Cortinarius identified. You can identify them if you have the appropriate resources and put in a little work. We're far from that here, but it's not impossible. It's difficult, but not impossible. So some things that we can do to ease that transition are using unranked names. So part of the problem here is that not all mushrooms are fully resolved into what we would call a species yet. They're a little bit different from each other, but they mostly look the same, maybe even can still interbreed with each other. This is what we call phylogenetic divergence. This brings up a concept that I call the Antlers of Evolution. Imagine that on one axis you have morphological difference, and on the other axis you have genetic difference. Some things are really clearly defined, they look really different, and they are genetically very different. And some things both look and sequence very similarly. So not every species is as clearly diverged from all other species, and that's an important thing to understand. So unranked names allow us to give names to taxa that don't qualify for what we normally would call a species. These are the clumps, clusters, swamps, bogs, mazes, and labyrinths of species. In other words, the messes. So a lot of the Cortinarius that look really similar and they all turn brown in age, that's a mess. That's a bog. We can make up provisional names, and that's something I'll talk about in a second here. So provisional names. Anyone know what that is? A provisional name is a name that carries no weight except for in your brain or by your definition. So this can be either really useful or not useful at all. But provisional names are, who's met Rod Tullas? Yeah, so he's this guy from the East Coast, probably the most knowledgeable person on Amanitas in North America. He studies Amanitas basically only. That's all he really is interested in. And he makes up provisional names all the time because he hasn't had the time to formally publish a name, but he recognizes that a mushroom is different. So he makes up a provisional name for it that allows him to refer to it without having to say Amanita species 06395, which is hard to remember. But he doesn't have to go publish it immediately because that's a lot of work. Provisional names can be changed when the actual publication time comes, but it's a way that you and I can communicate about them until that day comes. And the awesome thing about Rod is that he puts up good descriptions, basic descriptions, but good descriptions of his provisional names. So they're public. So they allow us to give names to things that we recognize in the meantime until something is formally published. But if they're public provisional names, they should also be publicly characterized. Otherwise, it becomes a total mess. So if you're going to make up a provisional name and use it, also make sure that people know what you're talking about by providing at least a photograph or a basic description of what it is that you're applying it to. Eventually, this will allow us to do things that are integrative, where we're not only just naming mushrooms, we're thinking about their ecology and evolution even, how so many of them were generated in one little forest. How is it that 15 different species of Inocebi can live on one Douglas fir? How do they all partition the resources of their host so finely that they can all make a living there? None of this is going to be resolved if we can't even name the mushroom that we pick up. So by learning to identify mushrooms better, we're going to be able to ask even more interesting questions, I think. And range expansions, if something grows into our range, like Blvd frostii might someday, unlikely, but a switch in host preference. If we see a mushroom going from native oak trees and living with eucalyptus trees, we're not going to be able to establish that that's happened unless we can call those two mushrooms the same thing, if we know that they're the same mushroom. The effects of climate change on population structures. None of that's it's happening right now, but we're losing information on it because we don't know what to call the organisms that are responding to it. So all of these big, maybe really important questions are related to the relatively unimportant question of what to call a mushroom. So it's important to start there and build up. So we need increased amateur access to a bunch of things. Herbaria. It's really hard to keep a bunch like thousands of dried mushrooms at home. I am lucky enough to keep mine at UCSC, but if I didn't have that, I don't know what I'd do. It's really difficult to do it on your own. So if clubs can provide access to herbaria at academic institutions or their own even, that's an important thing to give amateurs access to. Microscopes, expensive and not really a popular gift item. So if you can help people get access to microscopes, that's important. Technical literature, often expensive, difficult to find, and really not everyone needs their own copy. Club libraries are awesome and the FFSC has one. Engage it, find out what's in the library and use their technical literature. It's a great resource. Sequencing centers, this is the hard one. Sequencing is pretty cheap now. It's not that cheap, but it's pretty cheap. And amateurs can get their specimens sequenced if they have access to a sequencing center and a little bit of money to do it. At the next ministers' meeting, I'll be talking about the current proposal to sequence the mushrooms at the UCSC herbarium. We now have a cost estimate and a place to do it, and they're very gung-ho. They keep sending me emails asking when I'm going to send the mushrooms. And I said, well, we don't quite have all the money yet. But it's something that hopefully is on the horizon. What's that?
No, that's not true. We have the money we already allocated. You just haven't asked me for it.
It's a big sum, but it's something that I think the Santa Cruz Club is uniquely positioned to do and will be the first of its kind. Yeah, Milan.
Use the fruiting bodies as access to the Microflora.
Yeah.
What is your whole percentage coverage of the whole flora?
Of California?
Things don't flower in Europe.
Yeah.
Some of them never fruit.
Yeah. I mean, it's going to go on.
Yes, or is there any idea?
It takes, so let's see. I think in a study they did in Europe, they had to sample a site 10 times before they reached saturation in their curves, over 10 years. So not just 10 times, but over 10 years. That varies by size of site, clearly, and diversity of the trees, but it takes a long time to saturate an accumulation curve of species. We're not close in California by any means. It's going to be people like you and I putting in tons of hours in the field.
20-year project, okay.
Yeah, we'll see. 20 years would probably get us a long way there. So what can clubs start doing now? Running workshops on how to use Mushroom Observer. How to keep a species list. This is a very basic skill. It's not hard at all. But your name, the date, the location, and all the species you saw with notes. Encourage people to do that and have workshops to that end. Also encourage them to upload those lists to Mushroom Observer when they get home or to a computer. This is how we start building these databases. Photography. This is a bit of an acquired skill. It takes some time to learn, but it's really crucial for mushrooms. It's really important to learn how to take at least a basic color photo that you can communicate with, especially in cases of rare mushrooms or first reports from an area. We can have loaner cameras. The mushrooms that I photograph with a Nikon D80, which I bought a bunch of years ago for like $1,000. It's now like $200 or $300 online. A club, and I'm considering doing this for the UCSC Myco team, just buy 5 cameras that people can loan with tripods. Because yeah, the camera does matter a little bit in terms of how good of an image you can produce. And if you train people on the club cameras and they can take them out for their weekend, they can produce a bunch of images that maybe they otherwise wouldn't have been able to. And that gives us high quality photography to work with. Loaner microscopes, those are really important. The FFSC has access to the ones at UCSC, and we do microscopy workshops every year.
We have 2 of our own.
And we have 2 of our own. So there are, if you want to do microscope work, you can do it through the club, yeah.
Do you think, how useful do you think dissecting microscopes are?
They can be really helpful in preparing a specimen for the light microscope, for the compound scope, especially little mushrooms. Other than that, they can be really interesting and useful when you're learning your way around a mushroom fruit body. You can get really detailed looks. But I would say I used it a bunch when I was starting, and I don't really use them anymore. Yeah. Vouchering workshops. We did one of these at NAMA, but how to preserve your specimens. So both Ron and Henry did a great job. They gave me really nicely dried specimens. These things are not going to rehydrate. They gave me tags with all of the collector's information, the location, the date. This is beautiful. This is exactly what we need. Vouchered specimens. These are both, first records for the county. I guess one is a little bit earlier. So this is the first record, and here's the second record for the county. But the fact that both the first and second records are vouchered, that's awesome. That's amazing. That means we're doing good stuff. We're learning how to do this. And we know how to do this. This is not new. But to do sort of routine vouchering, where every foray that we have, where we have tons of mushrooms on the table, someone who knows what's common for the area and what's not goes through and picks out the uncommon things and makes sure that those get dried so that they are available for future sequencing or microscopic research. Because if you don't have a specimen, you can only hypothesize about it in retrospect. Getting people access to primary literature, we already talked about that a little bit, but that also means generating your own primary literature. Club can say, this year we're tackling those pesky boletes that have red stipes and yellow pores and bruised blue. We're going to figure out which ones we have in the county. And get together some sort of project and meet and make a publication at the end of the year that you put on the website. We have this new website to have these articles on. You guys can do this.
You know, I'm looking at this list, and it sounds like a job description for the science ministers.
Set up Club Herbaria. That's what I'm doing here, Phil. Yeah, I actually would love to. I think it would be, so this is a crazy idea I had while walking over to the Thai restaurant where we had dinner today. I was like, how cool would it be if we all sort of stuck our fingers in the pot and published a species together as a club, and we list every one of our damn names as the authors of the species. We could do it. There's no reason we couldn't. It would be awesome. I think we should do it just as an art experiment. So what else can clubs start doing now? Taxonomy-focused events, in addition to what we do at the fair. At the fair, I think we're doing public outreach. That's extremely important because that keeps the flow of people coming to the club, supporting us, getting out in the woods, finding new things. But we also need to do taxonomy-focused events for people who already know what they're doing. It's hard to do both at once. A lot gets thrown off the tables at the fair that's actually really interesting because we don't have time for it. It's about the public at the fair. So we need to have events like the Oregon Mycological Society does. They do a 3-day camp where it's all about vouchering and identifying and building skills. Finding out where you've been wrong all these years and where you've been telling people all these years and you were right. That's what the Myco Camp model is, and I really think we should do things like that more frequently. Adam and I have got some things on the docket upcoming this year that are going to be very much modeled after that. They're a little bit different to run, but they're still relatively straightforward.
Well, something like the key council that they have up in the Pacific.
Exactly, yeah. They meet every year to revise the keys that they've done. I realize I'm going over time a little bit here. I'm going to get Adam up here in a second, but a few more things. Adopt a location. Choose a spot close to home, make regular repeated visits there, make a photo and species list and put them on MO. This could be your yard. Darwin DeShazer has hundreds of observations from his yard on Mushroom Observer. It's awesome. He knows every mushroom that grows in that yard. Yep. All of these Agaricus bernardii are from the same spot in the ground over the course of 4 years. This is near my house on Laurel Street, and every time I walk by and I see they're fruiting, I go home and get my camera and come back and take a picture. So this is, I don't know, when I was a freshman, sophomore, junior, senior.
You should put little faces on them.
Maybe I will. But the great thing about adopting a patch is that this helps us get an idea of the variation that a single individual can do over multiple years in different climate conditions. Some years it makes 3 big fruit bodies, some years it makes 10 little fruit bodies. That's interesting data. We don't know how to interpret it yet necessarily, but it's interesting data. So I've chosen kind of a big patch, but you can choose a little patch or an even smaller patch. It doesn't really matter, just choose a patch. Smaller is more tractable, obviously. If you travel a lot and you don't really have a patch to call your own, adopt a group. Say Cortinarius is my group, or Amanita is my group, or Entoloma is my group. And wherever you go, pay attention to those in particular and become sort of someone that I can go to if I want to ask you, hey, does this mushroom look pretty different once you get out east? People who are knowledgeable with a single group across geography are really valuable people to have when you're compiling a Microflora. So I pay attention to the Entolomas. These are all pictures of Entolomas that I've taken, except for that one. That's in Australia, which I've yet to see. But it's the group I pay attention to. Even if it's a boring entoloma, I feel compelled to document it because I have decided that I'm going to pay attention to these. And it really takes, people who are deciding that they're going to do things exhaustively rather than just the pretty mushrooms to get a Microflora together. So, what makes Santa Cruz special? We're small. We have endemic habitats. There's some really cool mushrooms here. But we also have the FFSC. This makes Santa Cruz special. There's not many clubs like this. Noah has told you this, and he's been to many clubs all around the world. We not only have a really good history, we've got people who know how to throw a party. We're all going to have to stay fed if we're going to put this Microflora together. We've got a history of mycophilia. As I'm sure you all know, Mushrooms Demystified was written largely while in Santa Cruz. And we do pretty well for ourselves. We have some resources. We can do this. We can be sort of at the crest of this wave. So as the North American Mycoflora Project gets off the ground, I think Santa Cruz and its folks, both young and old, we have both people who can get out in the woods every single day and mentors for them. This is a really unique situation that Santa Cruz has. I think we can sort of be in the position to contribute a sort of special role to the North American Mycoflora Project, and I would hope that we find ourselves in that position through a little work in the next couple of years. So we can do this. Thank you for coming tonight. That's my talk. Adam's going to talk a little bit right after this, so don't go away. But yeah. Okay, Adam's going to give a quick rundown of our website.
This will be fast, don't worry.
Okay.
So basically what it comes down to is this, everything he's talked about, all this, it's been in discussion now actually for probably about 2+ years. Henry was one of the seeds of some of these ideas. Phyllis had conversations with us. And it just came down to this was the go or get off the pot moment, if I may say that. The way I describe it, we're currently in the renaissance of mushrooms in the scientific realm, and very few people get to live in a period where you're having a scientific renaissance. Obviously, this isn't like the discovery of electricity or a transistor or something, but it's a period that anyone that wants to can make their mark. If you follow what Dimitar is doing, he's at the end of the day just some guy. And he's publishing mushrooms that people have been arguing about probably longer than I've been on this earth. And he just basically put his money where his mouth was and went after it. In fact, he just gave us a new Amanita, Amanita Augusta. So you can all forget Amanita francheri because it's now Amanita Augusta. And he writes pretty good papers, and you can go read his paper. So to bring this all together, what did we do? You've got all this stuff going on, and he's talking about the Mycota of North America, which a lot of academics, a lot of talking. I hear they drink a lot. They're not getting a lot off the ground yet. So we said, okay, whatever. We're going to do it for Santa Cruz, and we're going to show them how to do their Microflora project. And so we started, and this has been an enormous amount of work. But the site isn't quite ready yet. But basically, this site is this Mycota project for just Santa Cruz County. And
so it's not live?
It is live because some of the people working on it, we're kind of distributed, so it has to be live so everyone can get in there and work. So yeah, you can go to it. It's scmicroflora.org. We've done a mountain of stuff. I mean, we've been out in the field with Justin making videos of how to ID mushrooms and Amanita videos, and we're a couple years in here, and we're starting to finally see some fruits of our labor. But just like we're starting to do stuff, but like earlier in the season, a professor at UCSC found truffles in her yard while she was doing landscaping, and it was just a lead we heard about. It was like, oh, okay, and I went up there, and sure enough, we don't know what they are per se. She ate some and said they tasted horrible. It was funny, she found them and ate them, then bothered to contact us to see if they were edible. Classic. We don't recommend that one.
She's a math professor.
Yeah, she was there when I was there. Yeah. So what we're going to be doing is
things going on as it pertains kind of the science of this. Amanita augusta, this just happened, it pertains to this county, Franchetti grows here. Laetoporus, we've personally seen it, it's starting to fruit. Miletus marshii is starting to come up. Big ones too. These are important. Miletus marshii, which is now about to be renamed, isn't well understood. So at the end of the day, as it loads, what this is, and it's navigable, is a comprehensive list of all the known genera of mushrooms that grow in this county. I can tell you this is large, having to sit and type it all in. So say you go into Agaricus. Here's a page all about the Agaricus of just this county. Currently we're saying there's 25 species. And they'll all be in here, because it all got to be entered some way. And here they all are. And you're like, oh, well, Agaricus augustus, for now. Go in, oh, here you go. Here's all the info we have, and nice pictures, and fruiting periods, and blah, 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 of Agaricus augustus as it pertains to Santa Cruz County. This is what the North American Mycoflora Project is attempting to do. They haven't even scratched the surface. So we're just going to do it for the county, and again, show them how to do it. Last thing, and I'll stop. So another way this can be a good resource is take Blvd, and a lot of people don't want to hear this, but it's coming. Blvd is being completely chopped up. Yeah. Currently, this county, per whoever you want to listen to, only has 3 Blvds, and one of them's been renamed. And a lot of people are like, what? So take Blvd appendiculatus, species formerly included in Blvd. Anyone know what Blvd appendiculatus is now? No? Bertroblvd persolidus.
What?
That's the butterbelie. And everyone's like, what the heck? Well, that's the point. Part of the point of this website is we're going to be covering all this stuff. So
It's about, don't everyone go saying that. It's not published yet.
it's not published yet.
It's about to come out, yeah.
Okay, friends, put your hand.
Yeah.
Put your hand